The last video we did was a key one because we went from primary to secondary structure of DNA. That is, we dealt with the double helix. And the two things that were really important there were the base pairing of hydrogen bonds and the major and the minor groove. Also, actually, I should add a third, the anti-parallel nature of the double helix. Now, this major minor groove idea and the base pairs is something I'd like to explore further because we're going to look at DNA structure from a different angle. And before we do that, let's just take a quick look at the actual structure of real DNA. This is a better description of it here because it shows you the full atom in a spherical ball and stick model here. Now what we can see here is the double helix. Here's one strand, here's another going in an opposite direction. Remember, nitrogen is blue, and the nitrogens are shown there. You can see those in the bases. And the phosphates are shown here with the negatively charged backbone. Now, what we can also see here is the groove is coming right down here. That is the major groove, sorry, that's the minor groove. And the major groove is coming around the back here and then coming up this way. So that's a little bit better view. You're going to do some homework on being able to sort that out. So let's move on and talk about base pairs. Going to get on to our next page here, and what I'd like you to do is take a look at these base pairs, and I would like you to, first of all, let's note that the position of ribose connectivity, I'll go put an RIB on there so you know that's ribose, or deoxyribose actually, is always in the same place like that. And what I'd like you to do as a little exercise is pause the video for a minute. I'd like you to identify the bases that we have shown here, and I would like you to identify any hydrogen bonds that you see holding these base pairs together. So let's pause for a moment and do that. Okay, this is going to take a little practice for you to identify these bases. Remember, you don't have to draw them from scratch, but I would like you to identify them. And the first one over here is an adenine, or technically it's an adenosine because it's attached to a ribose. And that A, we know because it's a purine with no oxygens present in it. And next door we have a thymine, or thymidine, and that is the pyrimidine that has an exocyclic carbon, that is a carbon outside the ring. Now over here we have a guanine, or guanosine, that's the other purine, and we have a cytosine, or cytidine, that is present base paired to that. So I hope you got those, and that's a good start. Let's go on and look at the base pairs, or the hydrogen bonding for the GC base pair. I hope that you would pick out that O, H, and N, and this N, H, and N, and N, H, and O there. Let's go ahead and put dot, dot, dot right there. All three of those, and you can sort out the donors and the acceptors there if you want as well. Now let's go back and do the same thing that we have over here for an adenine and thymine base pair. We have an NHO, an NHN, and I hope that you did not make this into a hydrogen bond because it's not. This is an O and an H, but it's an H attached to a carbon, and that is not a hydrogen bond. So what we have here is two hydrogen bonds between an AT base pair and three hydrogen bonds between a GC. That makes this one stronger because it's held together by more bonds. What else can we say about this? I'd like to point something interesting out. The ribose connectivity is not directly across from each other. They are actually at an angle, and that's going to be an important part that we talk about below. So what we need to say is that an A is always going to be matched with a T, and a G is always going to be matched with a C, and vice versa, T with A and C with G. So those are the base pairs. Notice you always have a purine and a pyrimidine together, and the hydrogen bonding must match. That's how you get correct base pairs. Okay, let's finish up down here below, and we're going to take these same base pairs and we're going to want to talk about the major and the minor groove. Now, most people could see the major and the minor groove in the previous view, side-on to DNA, but it's a little harder to see in the end-on view of DNA. And what it has to do with is what I just said, and that is the ribose connectivity. If we were to look down DNA end-on, we'd see just about a circle that's roughly 20 angstroms across, as we talked about before. And as I said before, the ribose are not 180 degrees apart. There's a small arc between them and a large arc between them. And what that has to do with is the major groove is formed by that large arc. That's why it's the major groove. And the minor groove is formed by the smaller arc. And what we can do is test you. I've changed the structure a little bit over here. Can you identify the major and the minor groove for the GC base pair? Take a moment and pause, and let's see what you come up with. Okay, I hope you were able to see that the ribose connectivity is key, and what that means is that the minor groove is always going to be found between the ribose connectivity that's small, and the major groove will be always found for the ribose connectivity that forms that large arc. Now, why is this important? It's important because DNA interacts with a lot of other molecules. In particular, it interacts with proteins. And the way it does so is, well, let's remind you that we have base pairs here, donors and acceptors. That's how DNA interacts with itself. How does it interact with other molecules? Well, remember, DNA has lots of donors and acceptors. So, for instance, we have a donor right here, an acceptor right here, and an acceptor here. Those are available donors and acceptors. And you can see a donor here, and an acceptor here, and another acceptor here. That's all available. And proteins can reach in and interact with donors and acceptors in order to read out the information that's found in the DNA. I'll try to show you some of that in our in-person sessions as we get together. So let's call it the end for this video, and we're going to continue on talking about base pairing in the next short video, which is a topic we call Chargaff's rules.